Thank you everybody for joining this webinar, and good morning, good afternoon, or good evening to you, wherever you are listening and watching. My name is Harold Bornfleth, and I'm responsible for the BESA product portfolio. So today I'm going to speak about exploring MEG and EEG brain connectivity in source space. For this special occasion, I ventured into the office for the first time after 8 weeks of home office. So please don't ask me what the plans in the office look like now. I'm also joined by my colleague, Jae-Yun Cho, who is not at BESA headquarters today, but he's in home office like most of us right now, and he will assist with questions and answers. So today's schedule is, first of all, I will give you a short introduction to the theory behind analyzing EEG or MEG brain connectivity. Then I would like to demonstrate some effects hands-on using the BESA connectivity software. Altogether, I expect this to keep us busy for around 45 minutes. And please, throughout the presentation, feel free to send in your questions. Simply use the available questions and chat boxes on your screen, and we will pick up all your contributions to the discussion, and Jayan and I will try to answer them at the end of the session. So what are we actually looking for in our MEG-EEG data is the first question. First of all, looking at normal brain function, brain regions with specialized neuron assemblies need to work together to achieve certain tasks, for example, multisensory integration. This can be achieved via synchronous oscillations, as, for example, reported by Singer and others in many publications. And then looking at pathological brain function, in these cases, network connectivity may be altered, which can also be investigated using MEG and EEG. So in order to find and analyze these phenomena, we need some ingredients. So first of all, we need time frequency decomposition. This can tell us at what time frequency oscillations occur, for example, relative to a stimulus. So you can see here a typical time frequency diagram where the time is shown from left to right and frequency activation is shown on the y-axis. The next ingredient we need is a connectivity measure, which allows us to extract the degree of functional or effective connectivity between channels. This could look like here. So the time frequency diagram now shows us at what time and at what frequency a certain degree of connectivity exists between a pair of channels. Talking about channels, another ingredient that's highly useful in this analysis pipeline is a disentangling of volume conduction effects from the actual synchrony effects that we are interested in. This can mean moving from connectivity on the surface, for example, on the sensor level, like in this example, to connectivity on the source level. And this is what we're going to look into more now. So let me have a look at, first of all, the first ingredient, the time frequency decomposition. Yeah, and why do we even bother with time frequency decomposition? So in case of stimulus-related data, we have the case that averaging increases the signal-to-noise ratio, but information that is not perfectly locked to a stimulus or to a trigger gets lost. So time frequency analysis can reveal relevant signal that's related to a stimulus, but is not perfectly time locked to the stimulus. And generally, we can say that for calculating connectivity between brain regions, we actually require a time frequency decomposed data. Yeah, and how do we get to this decomposition? In order to extract the frequency components that are active in a certain time window, I need to define a template, and this template will be convolved with my signal of interest. This slide here shows two example templates of the so-called Mexican hat wavelet type. You can see that the left template will pick up a higher frequency than the right one. So we can design templates that are optimized for any frequency, really. And another way of doing the same thing is using the so-called complex demodulation. And this method uses a sine or cosine wave that reflects the test frequency that I'm interested in and multiplies that with the data. And if this is in resonance with the frequency that's present in the data, then the result will be a beat frequency that is basically the difference between the actual frequency in the data and my test frequency. And this can be filtered out by applying a low-pass filter afterwards. And then we get our results for the time frequency decomposition in that way. So which type of time frequency analysis should I choose then? Yeah, there's always a trade-off between time resolution that can be achieved and frequency resolution that can be achieved. In complex demodulation, this ratio is fixed. So I decide beforehand if I want a good time resolution or a good frequency resolution or a trade-off between the two. But actually, in many cases, I maybe don't care about good time resolution and low frequencies, but I care a lot about this for high frequencies. And in wavelet analysis, these wavelets can be adjusted such that for each frequency that I'm investigating, an optimal trade-off can be achieved. At least that's the theory. So in wavelet decomposition, we have different possible choices for wavelets. The first one, and a very popular one, is the Morley wavelet. This is the formula for the Morley wavelet, which you don't have to memorize, but there are two important parameters here. The Morley parameter oscillation, which is the wavelet parameter, and then the second one is the length of the wavelet in units of this sigma that reflects the length of the wavelet of each wavelet cycle. And then another type of wavelet that we've also implemented in our tools is the Mexican hat wavelet. This uses the second derivative of the Gaussian, and here there's only one parameter, which is the length of the wavelet again. These are some examples of wavelets. So you can see the oscillation parameter from left to right. And obviously the oscillation parameter has quite an impact on the shape and the look of the wavelet. So you can see the oscillations equal 3 is a much different shape than oscillations equal 7. However, the length is also plotted here. So this was the length equals 3, and down here we have length equals 5. And that effect doesn't seem so strong. So usually length equals 3 should actually give us a good enough result. Also, what's important in wavelets is that we can define a real part and an imaginary part. So here we see an overplot of real and imaginary part of a Morley wavelet. And on the right-hand side, the same for a Mexican hat wavelet. So that's fine, but then what good does the imaginary part do us? And this leads us to the next ingredient that we want to apply in our analysis, which is the connectivity measure. So looking at connectivity measures, many of them are phase-based connectivity measures. This means they use the relative phase between two channels in the connectivity calculation. And in order to do this, we need the real and imaginary parts of the wavelets. So typical phase-based connectivity measures are coherence, imaginary part of coherency, phase locking value. And then there's a different family of connectivity measures, the Granger causality-based methods. And here members are the Granger causality itself in the frequency domain, the directed transfer function, and partial directed coherence. There are also others, but these are the ones that we currently have implemented in our software solutions. So looking at phase-based connectivity measures, coherence. Coherence is maybe the best known connectivity measure. This describes the amount of in-phase components in two signals at a specific frequency. This XS, XY, this is the cross-spectral density between channels X and Y. And the chart at the bottom here shows an example for how different relationships between two channel signals will affect the calculated coherence. So we can see 5 example trials, trial 1, 2, 3, 4, 5. And here plotted is the amplitude of channel X and Y, symbolized by the length of the arrow, and the phase angle between them at the different trials, indicated by the angle between the two. So you can see that in this scenario, in the first row, whenever the amplitude of signal of channel X gets lower, also the amplitude of channel Y gets lower. The phase relationship is always the same. So we have an ideal correspondence between amplitudes and phases, and this will lead us to a final coherence value of 1 between them. And in the second row, we have another example where there's a correlation between amplitudes and phases. For example, the phases are always here in the first quadrant between the two, but it's not fully stable over time. So there is a correlation, and that means the coherence is there, but it's not 1, it's smaller than 1. And finally, in the last column, last row here, we can see a random association of phases and also amplitudes between them. And in that case, we would measure a coherence value of 0 using this formula by Rosenberg and others in 1989. Then we move on to imaginary part of coherency. So coherence was great, but one big problem was that any effect that was simply due to volume conduction, for example, if you think of spreading of a brain signal that can spread over several electrodes, this will automatically come up as a perfect coherence, when in fact there's nothing coherent between different brain regions there. It's just a coherence between channels that measure the same thing. So then the imaginary part of coherency was introduced to exclude that phenomenon. So coherent sources with 0 phase lag would no longer result in a value of imaginary part of coherency, basically by using the imaginary parts of these calculations. Another thing about imaginary part of coherency is that it has a also has a characterization that it can tell us about signal flow between sources. So if the value is positive, that means that time course Y would be earlier than X, and the information flow is then suspected to be from Y to X. And then we have the phase locking value. This is another measure which discards the amplitude information altogether and just looks at the phase relationships. This again is a non-directional connectivity measure. So we can measure the amount of coupling, but we don't know which way the information flows. Which brings us to Granger causality-based measures. Granger causality states that if past values of time series Y contains information that helps in the prediction of time series X, then Y is said to cause X. So this is really a different approach to the connectivity problem. The measure is the logarithm of the unexplained variance from X's autoregression divided by the unexplained variance in the joint regression. This sounds really complicated, but just imagine that the joint regression is a better predictor than the autoregression. For example, this could mean, if you have a smaller sigma 2 than sigma 1 because there's less unexplained variance in the joint regression, then the Granger causality will rise in this case from this formula, meaning that Y helps to predict the signal of X. So these are the formula for the frequency domain. Now this gets a bit more complicated. I can maybe skip the explanation, but you can still see in the final formula familiar cross-spectral density matrix S, XX in this case, and this is which then gives us the self-spectrum here. The other two measures left to discuss are directed transfer function, which was introduced to reveal indirect information transfer between signals, and which uses the transfer function matrix H which compares transfer from channel J to channel I with transfer from all channels to channel I. And then there's also partial directed coherence, which looks at only the direct interrelations between the signal. So basically, directed transfer function includes direct and indirect transfer, but cannot differentiate between direct and indirect connection. And PDC, on the other hand, gets rid of indirect transfer and keeps only the direct interrelations. Now, about implementation of these concepts into the BISA software. So BISA uses the non-parametric approach. This means that we do not require to determine the model order for the autoregressive model, which is a good thing. But nothing comes without a trade-off. So here the trade-off is that we need a smooth shape of the frequency spectrum. So in order to compute these Granger causality-based methods, we should use the entire frequency axis or at least a large frequency range for best results. So with this as a given, we can move to estimating the cross-spectral density matrix, then factorize it, compute the transfer function, the noise covariance matrix, and then we have everything together to estimate the Granger causality-based connectivity measures. Okay, but we're not quite finished yet. The third ingredient that I would like to point out comes because of the issue of interrelations of channels. So we were talking about interrelation of channel I to channel J, then there was channel K, etc. So ideally, we would really like to investigate just the information exchange between neural generators in the brain. Not so much what's going on the scalp, where channels I, J, K may show us exactly the same brain activity, just propagated to the scalp. So signals are usually collected on the scalp in the EEG case or some distance away from the scalp in the MEG case, unless you use OPM. The skull acts as a strong insulator for electric currents, especially in the EEG case. That affects EEG, and spatial filtering can be used to transform the data from sensor space to source space, which is what I would like to talk about in the third part of this presentation now. Yeah, so what about source space? Well, if you think of what's actually going on before we measure the EEG or MEG signals, there's a neuronal current flow. It flows perpendicular to the cortex and it creates dipolar fields. The orientation of these currents can be radial with respect to an approximated sphere representing the head surface, or they can be tangential, or there can be something in between. So if we look at a radial current, it creates a widespread smeared voltage topography over the whole scalp. You can see on the left side here a topographic map of this radial source here. So you can see that several electrodes have a very high negative signal, and then there will be some positive signal on the other end of the of the scalp. And you can see on the right-hand side how that looks in this electrode set. So the negative maximum will actually be over the activated cortical sheet here. And the maximum of this signal is spread over around about 4, 5, or 6 electrodes in the center. If we now look at an activity in the fissure that creates a tangential current, now the sensor topography has two maxima, one here at the front and one at the posterior aspect of the scalp. And these signals will be 100% correlated or 100% anti-correlated, if you like. But actually, if you look at the electrode distribution here, none of the electrodes that show the highest signal are even close to the location of the neural current in the brain in that scenario. That's just the reality of measuring signals from a distance. Looking at the same thing in the MEG, it looks slightly more promising. So in this case of a tangential current, the MEG has an advantage, and it would show you a more focal maximum roundabout over the area where the neural current was. But even in the MEG, this is spread out over several sensors here. So if we know about the underlying generators from source analysis or from the literature, we can model each brain generator by one source. And then we can apply a spatial filter to transform the signal from the scalp, from our many sensors on the scalp, to the brain, to much fewer sources in the brain, into a so-called source montage. So the source montage allows us to calculate the response of the brain regions to the stimulus on the individual trial level. So say we have a source model like this on the left-hand side. This can have dipoles, like in this example. You can have regional sources that just summarize anything in a certain brain region without taking care of the orientation. Or it can also have beamformer virtual sensors. And this source montage can be applied as a spatial filter to the original raw data. And if you look at the individual trials, then you can actually see how much current in each individual trial originates from a certain brain region. Yeah, and then we can use these brain generators, these virtual sources that we have now in the brain, to calculate our connectivity between them. So the concepts that I mentioned in these three ingredients, they are actually present in several toolboxes. And yeah, I would now like to demonstrate the pros and cons of the of these approaches using the BESA research pipeline. And to do this, we need to follow 5 steps. So the first one is in our BESA research, we load the raw data. We select a montage or source montage of interest. That's step number 2. Then we need to define the trials that we want to work with, and this is done via the ERP or ERF paradigm. We then define a condition there, and this condition will tell us which trials will be analyzed. Then we launch the BESA connectivity program, which then takes us into the workflow to compute time frequency transforms. That's step number 4. And finally, to continue with the connectivity workflow. Okay, so let's move into this. So this is the display of the BESA research program, and I have chosen an example here that has 128 channels. The data were already epoch together, so this is why we see some jumps here every few seconds. That just means that trials were added, and these are the raw data, but they're already epoched. And you can see down at the bottom the trigger numbers. 31 was a stimulus, 41 was a reaction. So the whole experiment was about error-related negativity. There was a visual stimulus, and the subject had to react. So there was this task associated with the stimulus. The subject had to react by mouse click, and there was either a correct or a wrong, erroneous answer. And these were then assigned different trigger values. The ones I think followed by correct responses are 31, and another trigger was assigned to stimulus followed by a wrong response. Good. So this is the raw data, 128 channels, and now we can transform this into source space. And yeah, this has been prepared. So we have a source montage that we call ERN, error-related negativity, with 11 sources, 11 spots in the brain that we're interested in, where we want to find out what the activation was. So just a brief look at where these sources are. So I open the montage editor here. And I can go through the different sources. So the first one is the primary visual cortex. Then we have lateralized visual areas, left and right. We have temporal lobe sources, left and right. And then motor cortex, left and right, and also cingulate anterior and posterior sources. And there was also 2 sources put into the eye just to cover up or to catch any eye activation that might distort these signals in the other sources. So this is the source montage, and you can see how the single trials are now transformed to the source montage here. So next step we need to do is that we want to define the paradigm for this. So the ERP module in BESA opens up this paradigm file, and we have 4 conditions defined here. The first one is a stimulus followed by an error, followed by an erroneous response with 201 events. And also we're interested in the stimulus followed by correct response, which is 307 cases here. Right, and just have a look at the epochs. So basically, we always like to have relatively long epochs because that gives us more wave or room for maybe post-average filtering if we need to. We have filters that we set up. Basically, we've only got a high pass set up here. And then there's the artifact scan. So this artifact scan allows us to get rid of bad trials. So you can see the whole experiment kind of sorted by channels in the rows and trials in the columns. And the bad trials are all sorted to the right. And I can move the slider to include or exclude more or less trials. So yeah, so now after excluding some, I'm left with about 80% of the trials, so that's fine. And now I want to do my time frequency analysis for these trials, for the 2 conditions. So let's move on to coherence, and here we can select our conditions of interest. So one I'm interested in is the stimulus followed by erroneous response. The other one is followed by the correct response. And I can use this Start Connectivity button to now move to the BESA connectivity module that will do the rest of the number crunching and display of the results. The first thing is that we export the data in the epoch format, a very simple binary format that actually is open, and we also offer it, for example, via MATLAB scripts. And now BESA connectivity starts, and I can now go straight into the workflow of time frequency analysis. So the left-hand side shows me the progress through this workflow. The right side shows me what I can influence in the workflow. So first of all, I need to just select my method for time frequency analysis, and that can be complex demodulation and or wavelet transform. So I'm going to check both of these. This number of conditions and the names have been pre-filled. They've been passed on from BESA research. So I can press the next button. And the next step is to actually load the data into the workflow. And also the file names are already pre-filled. So I can just press OK, or I can advance using the space bar on the keyboard. And now I get a little summary of the data here. So yeah, the number of trials, the number of channels. This is my source montage, so 11 channels, and sampling rate, etc. And move on. And now, yeah, if I hadn't done an artifact scan yet, I could now also go through the single trials and identify any rogue trials and uncheck them here, so to exclude them from further analysis. But I don't need to do this now because I've already sort of cleaned my data in that way. And here I can define the cutoffs for frequency. I'm going to work with 2 to 50 Hertz, so I don't want to include the 60 Hertz mains frequency, but I want to have a broad frequency range. And I'm just going to stay with 3 oscillations because that should give me a bit of a smoother frequency spectrum. And here we have some other settings for complex demodulation, which I'm going to leave as they are, and then press next and start the time frequency decomposition. And that should be quite fast. Yes, here's the result. Now I can see the so-called temporal spectral evolution of the data over time. That means that I can see the change in activity compared to the baseline. So for each frequency, I can see here there's a baseline interval, and then afterwards does the activity increase or decrease. That's usually more telling than just showing the straight amplitudes, but I can also switch to straight amplitudes with these different buttons here. So let me check a different channel. So yeah, on the left-hand side here, I can see the average waveform. On the right-hand side, I can see the actual TSE display. So this is my virtual, not virtual, this is my visual right area. And scaling it up a bit, I can see there's some synchronization happening at low frequencies and some desynchronization happening at around about 11 Hertz. So I can also change the method between the time frequency decomposition. So let me switch to Wavelet. So I can see with the Wavelet, this actually comes out better. So I can see more desynchronization here in the Wavelet analysis. And yeah, let me also switch between conditions. So this one was the error condition. And looking at the, for example, the singulate signal here, posterior singulate, very strong synchronization, quite late, around about 300-400 milliseconds after stimulus. And if I switch to correct response, then I can see there's hardly anything there. So it's really a huge difference between these two conditions, especially in the singulate signal. Okay, so that was my time frequency decomposition. So let me just finish this workflow and save it. Save it in the ERN. So, and usually I would give it sort of a name by two conditions, source montage, complex demodulation wavelet with parameter 3, and save that. And now go to the connectivity analysis workflow. So you can see we're back in the workflow selector and switch on the connectivity. And now I'm going to use this time frequency decomposition to crunch the numbers for calculating connectivities. So first of all, we've got coherence. We want to check imaginary part of coherency. And I'm also going to check on Granger causality. But I'm going to leave the other ones because we don't really have that much time. So I'm just going to show the highlights, hopefully. So next, and select the project that already is should have been selected. Oh, okay. I actually made a new group. So instead of giving the project a name, I gave the group a name, and then the project was just called project. Okay, but it's not lost, it's there. And let me then go further. So here again, I see the overview of my data. I see the time frequency decomposition again, and here I can now adjust some parameters for my Granger causality methods, but I'm just going to stick with the defaults here and run the connectivity analysis. So this is now going to take a few seconds. It's running through the two conditions. It's running through two different types of time frequency decomposition, and also investigates 3 different connectivity measures. So altogether, that is 3 times 2 times 2, so 12 different types of calculation that need to run on this data set. And of course, each data set has between 100 and 200 trials, so it takes a little time, but here is the result. And the result, first of all, is a display of the coherence, because that's the first method that I selected. And it shows me all channels by all channels, so it's the full matrix of connectivities. And by default, it looks for the highest value and shows me this in the bottom. So at the bottom, I can see the two channels that have the highest coherence and at one point, and I can see also the names of the channels. Right. First of all, I'm going to hide the diagonal because that's just the self-spectrum and is not relevant for my coherence between channels. And what I can see is here that I have some quite high coherence in the higher frequencies, and this is something that is due to volume conduction. Even though I've now actually moved from the sensor into source space, some noise will be enhanced in the same way. Noise that could not be modeled by the sources will be modeled in the same way by different sources. So that'll create some artificial coherence in the high frequencies that is not probably a real effect. Let me see what this is like for the wavelets. So switch to wavelet type. And the wavelet is a bit less pronounced, but it's still also a factor. I am in the error condition. I'm gonna look for the imaginary part of coherency. So this actually now gets rid of my high frequency noise. So if you compare back with coherence, okay, where was I to begin with? I was somewhere here. Go to imaginary part of coherency. So this is one good thing about this imaginary part of coherency. It really takes away these zero phase lag connectivities, which are possibly just an artifact. Yeah, and now we can see there's one channel with a very high connectivity. This is my singulate posterior source between singulate posterior and singulate anterior. And I'm gonna just click into the maximum here and show the 3D mode. So in the 3D mode, I can now see the connectivities between all channels graphically more clear, and particularly for the time and frequency spot that I've selected right now. Yeah, this is my error condition. I can see the singulate is very active here, so it has also lower the threshold here for showing the signals. You can see that singulate dominates this and is connected at low frequencies to a number of other sources. And let me switch condition to go to the correct response. And interestingly, there's hardly anything going on now. So yeah, I can move back a little bit. So there's a little bit of connection, but compared to what I saw in the error condition, I've got a much stronger response in the error condition, especially at later latencies at these low frequencies compared to what I'm seeing in the correct condition. So let me go back to the 2D mode and have a look at the correct condition again, and something in the, there's something here that also is quite interesting, a bit earlier latency going on between the lateralized visual right area and the motor cortex. And let's have a look at the 3D mode for this. So here we can see that's a different network now. So here we have the visual right area and the motor cortex right, and also the main visual. So they're connected in some way at this earlier latency at a higher frequency, which is about 10 Hertz here. And we can also have a look at the Granger causality now. And yeah, Granger is, there's not much can be seen. So let me go back and scale this up in the 2D mode, so I can scale this, and I can change the color map to show this a little bit better. So change to Viridis and scale up a bit more. Yeah, now I can see there's something happening also in the Granger causality here that emanates from the background, and I can have a look at the 3D mode again. For this, I just need to lower the threshold accordingly, and then I can see what's going on. So lower the threshold, scale up. Yeah, now I can see, yeah, there's also in the Granger causality, I can see this effect of the network. Maybe make the transparency a bit more. And I can also see who's the driver. So I can see that the information flow is from the prime visual areas to the right lateralized and then to the motor cortex. And if you look on the other side, this is the connectivity back from motor cortex to visual, right? And there's hardly anything happening here. So it's value of 0.016, and at the same time here it's a factor 10 higher. So obviously it's completely directional. Yeah, and we can also investigate this a bit further by, for example, averaging over the frequency. So I can now change my frequencies and look at so the same latency, or I can look at the latencies and see what's happening between 10 and say 19 Hertz. Yeah, so obviously for this source, the highest connectivity is just where I picked it at this frequency range. And then I can see, so, okay, these are very small connectivity. So there's mainly this thing going on in the right part here. Again, in the correct condition, in the error condition, there's hardly anything happening at this frequency range. But in the error condition, I can go back to lower frequencies now. So if I look at frequency range, say 2 to 5 Hertz. Yeah, okay, in this particular source, there's not much going on, but you can see this huge stuff in the singulate. So there's a completely different network happening in the low frequency range compared to what's going on in the high frequency range. And this can be investigated very quickly, very interactively using these tools here. Right, okay, so this is just a little glimpse into the, so back to not averaging over frequencies. I can now finish the workflow. This was a glimpse into the how we could look at this error-related negativity. I'm just gonna keep calling it project. And I can also have a quick look at an MEG example. Maybe I've only got 5 minutes or so, but let me try and do this. So if I go to another example, this is a more simple experiment. This was a visual flow field. So there was a trigger 1, that was a flow field moving towards the spectator, and trigger 2 is just a static visual stimulus appearing. So we also have a source montage for this. So there was 4 dipoles that were, show you a little diagram here. So the 4 dipoles are primary visual cortex, still primary, but slightly lateralized for the number 2. And then dipole 3 and 4 are in lateralized areas that are actually in the MT regions, which should be responsible for motion related things. Okay, and we can already see how these single dipoles pick up signal after the trigger happens. So obviously this dipole model is quite sensitive to what's going on. And again, I go into the ERP. I've got my 2 conditions, motion and static. Artifacts have been rejected and go straight to coherence. So again, exporting the binary data. And now we are starting the coherence module with this data here. And I'm just going to work with Wavelet now, since that seemed to be more informative, and open the existing data. and I'm gonna cut off at 80 Hertz, and again work with the 3-3 combination. Run the time frequency analysis. So now I can see there's mainly synchronization happening in the channels, more pronounced in the right than the left hemisphere. Finish this workflow and call this project Visual Motion and start the connectivity analysis. And yeah, just gonna work with the same 3 as before. Open this project Visual Motion, it's already pre-selected. Go through the workflow and run the connectivity analysis. And that should be quite fast because it's only wavelets to compute. Here we go. So that's the coherence again. Let me go straight to Granger causality and change the color map again. So here I can see there's this seems to be the strongest connectivity that was found, and in the 3D mode I can look at it. So yeah, indeed there's mainly connection from primary to the D3, which is in the empty cortex, which is true for the motion stimulus, and for the static stimulus I have nothing like that. So I've only got a little bit going on at very much lower frequencies. So for the motion, I think I was at 12 Hertz here in my maximum. Yeah, and I can play with the, can scale up a bit. I can play with the thresholds to see at what point do the other ones join. You can see there's also connection to the left lateralized, but that's slightly less strongly pronounced, but still is there. Good, and yeah, then of course we can export these. There's other things we can do, for example, create videos. So yeah, we can export a video, but I think this is also part of some YouTube videos, and we're running out of time. So yeah, maybe you'll have a chance to explore it yourself. And I think the video function is very nice. Also, all the results can be exported into ASCII data and can be loaded into MATLAB for further analysis. And I think now I will go back to the presentation, mainly to thank the other scientific contributors to this project, Robert Spangler, Jaehyun Cho, Avinash Pant, who were very instrumental in putting together the Bezier connectivity, and Michael Shirk, Patrick Burke, my late colleague Dieter Weckeser, and Nicole Ille, who contributed to the source montage mechanisms and formalisms.